Well, thank you, Brian. It's really a huge pleasure to be here. I'm so pleased to be invited and to tell you a bit about some of my more recent thoughts about reproducibility. And thank you very much. I got really interested in reproducibility fairly near the start of my career when I was finding peculiar things in data that I was getting. I remember Fiona's story about p-hacking. It wasn't that dissimilar in my particular life. But I suppose I became most heavily associated with the idea in 2015 when there was this Academy of Medical Sciences Symposium in London, which was supported, interestingly enough, by three major funders in the UK, the Medical Research Council, the Biology and Biotechnology Research Council, and the Wellcome Trust. And they were getting concerned about the fact that a lot of the research that they were funding appeared then not to be useful for drug translation and so on. And we really had a two-day meeting where we covered many of the same sorts of issues as we are covering here at this conference, but we came to the conclusion there is no one major cause and there's no one major solution. So the sorts of factors that we thought were involved were ones that are in this middle box, so things like data dredging or p-hacking and so on. And we came to the conclusion that to solve these problems, we would need both top-down change, so in terms of incentives for scientists, and also bottom-up change in terms of more training so that people could understand the methods better and so on. And what we missed, though, I feel now, when I started thinking about it further, were what I call the cognitive constraints that make it hard to do science well. And that's what I want to focus on today, because I think it's a part of the puzzle that isn't talked about very much. And yet, as a psychologist, I'm very interested in how people think and reason. And I think we do have some insights from research in psychology into why we keep persisting in not necessarily doing things right. I had to give a much longer talk just a couple of months ago, and I covered 5 different factors that I think are cognitive biases or cognitive constraints that I think lead to research not being as good as it can be, which are listed here. So seeing patterns in noise, systematic misunderstanding of probability, the need that we have to use narrative to organize our thoughts and explain them to others, and then this one, asymmetric moral judgments and confirmation bias. And I'm going to focus here on those last 2, just because I had thought, shall I try and cover everything very, very fast, or shall I just cope with a couple of things where I can tell you in a little bit more detail? I'm rather sad that Danny Kahneman didn't give his talk, because I'd rather hoped he was going to cover number 2, but never mind. So let me just start with the idea of p-hacking, which has been talked about a lot at this meeting, but not really, it's just been assumed that we all know what it is and have some idea about how it comes about. I think it's very interesting to me that in many fields, and certainly in psychology, it is still really quite normative, that you see papers all the time at conferences, you get things sent to review, where it seems to be the case that p-hacking has been used. And the question is, why is it so common, given that people have started pointing out the limitations of it? But let me just say what I think happens here. This is just an illustrative example for those of you who are not so heavily into this. This is just a sort of theoretical illustration using the sort of thing I am interested in, being a neuropsychologist. So I might be interested in laterality or handedness in children with developmental disorders. In fact, I am. I do a lot of work on that. But let's suppose I think there might be an association between attention deficit hyperactivity disorder and handedness, and I have a nice big sample of data with a large population, and I start to compare them in terms of whether handedness differs in the ADHD children versus typical children. And I'm a typical psychologist. All I really want is p is less than 0.05 to emerge from this comparison. And I do one contrast, and I'm gravely disappointed that p, alas, is greater than 0.05. But then I notice and I think, actually, I've got some very old children in here, very young children in here. In the younger group, there's a sort of glimpse of something on the horizon. So I think, right, well, maybe this is going to creep into 0.05. And let's suppose it does. And I get rejoiced and I get excited, and I think, let's go away and publish it. And I decided that I'm not going to mention the older children because they're just going to sort of confuse people. And so I say, well, here I am. I've got a result, p is less than 0.05. So this is classic p hacking. And of course, the problem here, which I think people have great difficulty grasping, is that this is the wrong statistic for this situation, because the statistic for this situation is not what is the p-value attached to this end node here. It's what is the likelihood that what is the probability that any end node would come out as significant. And that, of course, is greater than 0.05. In this case, it's 0.1. And to get a proper p-value, you'd need to Bonferroni correct it. And you can extend this, as people do, and you can say, well, I've got two measures of handedness. I've got a skill measure and a preference measure. And the skill measure is looking really promising. And then you can say, well, I've got males and females. So these young females on the skill measure, I mean, this is looking like a really good association. And as you can see along the bottom of the slide, this p-value that you need to really be using, because you should really be thinking about the whole constellation of possible results, is getting bigger and bigger. And by the time we get to the end, where we've, by magic, found that if you take young children who are female and on a skill measure and who are urban rather than rural, you achieve p less than 0.05. And you think, yes, I can now publish this and I can make up a theory as to why this should be the case. So that's hypothesizing after the results are known. But of course, by this point, I've got more than a 50% chance of finding something at the end of one of these nodes. So this is the sort of garden of forking paths being illustrated, which I think Andrew Gelman was the first person probably to apply that very nice phrase from a Borges novel to this sort of situation. So that's your p-hacking. So why do people do it when it's not right? Well, it's certainly not approved of if you talk to the general public. So if you ask people, as they did in this study by Pickett and Roche, whether it's acceptable to do this sort of thing, and they give people fictitious scenarios, and here's a doctor inventing a drug, and they decide that they are going to only report the results that came out as significant, you can see on this right-hand column here that people don't like it. They think it's morally unacceptable. Indeed, 63% of them think you should be fired. It's not as bad as if you actually falsified your data and made stuff up. But if you did this sort of omitting crucial information, the general public think you should be fired, possibly receive a funding ban, and some of them think you should go to prison. That's perhaps a little extreme, but it's not approved of. So what about the psychologists themselves? They're doing this, and are they not feeling bad about this? Well, there's research on moral judgments, which I think gives us some insight into what's going on here. And that is to do with the fact that, as you will have seen in the previous slide, although people didn't like you omitting data, they liked it a little, it wasn't regarded as quite as serious as actually falsifying stuff and fraud, making stuff up. And there's been research in a sort of parallel universe on negotiation. There's this sort of business studies type research where you give people, again, fictitious scenarios. So you've got a used car salesman, and he may either, when he's trying to negotiate with you about the price, he may lie and tell you that this car is a certain age when it's much older, or he may just omit relevant information, like the fact that it hasn't got a spare wheel or something, or he may say something that's true but in a misleading way. And that was known by people in this field as paltering, and it's what politicians do. It's when somebody asks you a question and you don't answer it with a falsehood, but you answer it in a way that omits something important, and it's intended to deceive. So you might have somebody say of the car, the customer asks you, well, does it always start properly? And you say, well, I've had it now for 2 months, and every time I tried to start it, it started. But you don't actually mention what happened before then, for example. You just tell them what the crucial bit that seems to fit with what they want to know. And the interesting thing is that all of these are regarded as dishonest, again, by your general public. But the paltering is regarded as not quite as dishonest as omission or lying. So there's a sense that this isn't quite so bad. You're somehow not being totally dishonest. And I would argue that p-hacking has features of paltering. It doesn't involve changing anything. You report, and indeed, people are rejoiced. I mean, they get this output from SPSS, and it says, p is less than 0.05, and they go, yay! And there it is in front of your nose, and you're just going to report that. And if you don't understand probability, which a lot of people don't, this may seem quite innocuous, because you think these statisticians that talk about Bonferroni corrections, they are just killjoys. I mean, they just out there, they just want to make your nice study go away. This lovely result, they're going to grasp it away from you by insisting on a Bonferroni correction. And people seem to recognize that it's not ideal. And there's this wonderful quote that I wish I knew the attribution. I think it's Uri Simonson, who said that if you asked the average psychologist, do you do this and do you think it's bad? People will say, well, it's not ideal, but they think of it more like jaywalking, whereas they should be thinking of it like burglary, because it really is quite serious in terms of its potential to really give you false results. So I've got very interested in this aspect of why people persist, and I think it's that many people really don't regard this as a serious problem, and they think it is just these pedantic statisticians who are trying to spoil their fun. And that therefore that would be something that potentially could be solved with a degree of education. But it seems to tie in with the idea that we regard errors of omission as less serious than errors of commission, even though the consequences might be as serious. I want to move on now to say something about confirmation bias, which you'll see how this comes into the position. It's another sort of cognitive characteristic of people, but if you put it together with p-hacking, you get into really serious trouble. But let's look at confirmation bias on its own first. So it's basically, it can operate at various points in the research cycle. I'm going to focus on an aspect of research that isn't much focused on, which is how we select other work to cite when we write our introductions to papers, for instance. And I think virtually everybody in this room who's ever written a paper will be in the position of knowing very well that what you do, typically, is exactly what is described in this wonderful little cartoon. You sort of think, well, there's two sides to this question. I'm going to really try and find some evidence. And you go onto Google and you go, Google, or you might, if you're a scientist, go onto Science Citation Index, but you go, blah, 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 and then you find a paper that says exactly what you want to hear, and you go, jackpot, you don't look any further. There you are, you've got what you want to cite. And again, we ought to probably feel bad about this because it's a very biasing sort of thing to do, but it's very much how we're wired up to think and not to go around. In fact, we would suffer from terrible information overload if we processed everything that's out there. So we're very much people who we're certainly not behaving like Popper would like us to behave, trying continually to disprove our hypotheses and our beliefs. Life would get very, very tiring if we did that. Instead, we tend to be looking for supportive evidence. Now, I want to sort of use myself as an example because it's very easy to point the finger at other people, but I think that because I'm saying these things are generally human things, I want to illustrate that even with the best will in the world, one does this. So here's a personal example of suppressed memory for relevant research when it doesn't fit. So one of the things I work on is children's language disorders, developmental language disorders. I'm very interested in genetic factors. So I've done twin studies on this, and I've given lectures on the genetic basis of developmental language disorders, where the main interest is in comparing in monozygotic, I don't know if I can get this thingy to work, no, never mind. The column saying MZ is the agreement between two members of a twin pair if they're monozygotic twins as opposed to dizygotic twins. And the prediction from a genetic model is that identical monozygotic twins will be more similar to each other than dizygotic twins who have only 50% of their segregating genes in common. And here we have 3 studies, 2 from the States, one British one I did, coming to pretty similar conclusions, all quite small sample sizes, but these are hard kids to recruit, but pointing to really to quite a strong genetic influence on developmental language disorders. So I would go around giving this lecture saying, look, we've got all this evidence, it's a highly genetic condition, highly heritable. And then this study came along in 2005, and it looks completely different. So there's not really much evidence, well, there's no evidence for any genetic effect here, and indeed the agreement between 2 members of a twin pair is quite low. And I had found, I suddenly realized that although I had known about that study, when I was preparing talks and things, I just never thought to put it in. I sort of managed to mentally suppress its existence. And it was only when I had to do some more systematic review that I sort of came up against it again and thought, oh yeah, that's that study by my PhD student. She wasn't at the time working with me, I have to say. She had moved on to work with Robert Plomin, where she did this study, but nevertheless, I knew about it. I mean, but somehow I sort of... So, I mean, we do this, and it's really quite serious. Now, there's even worse things, and I'm going to use here an example from somebody else's paper without naming them, because I really don't think naming and shaming is appropriate, because this is so common. But this is one which I sort of came across because an MSc student was asked to do a journal club and to pick an article, and they picked this article and had the misfortune to give me an article where I read the introduction and went, oh, no. So this was because this is a paper on autism and cerebellar neuropathology, and in the introduction it says, cerebellar neuropathology commonly occurs in autistic interviews. And I just said, really? That's not what I thought from what I know of that literature. And I'd been to a meeting on neuropathology where this seemed to be something that was mooted, but was highly controversial. But they've got all these little references. So I looked at all the little references. Most of them are, well, there's 2 basic review articles that cite the very small number of studies and come to rather different conclusions. And one of them comes to the conclusion that there's far too many confounds to conclude anything. But then there's these MRI studies. So brain imaging report that autistic children have a smaller cerebellar vermal volume, a bit of the cerebellum, in comparison to typically developing children. And so I thought, well, maybe I've missed all the MRI stuff. There may be a systematic review. Let us see if there's a systematic review. Note that this study was published in 2013. Here's the systematic review, which was published in 2018. Note the dates of all those studies in the systematic review, which are prior to 2013. Only one of these studies was cited by this person, this one in the red box. But notice, it doesn't find smaller cerebellar volume, it finds bigger cerebellar volume. It's the only study that finds any difference in cerebellar volume, and it's in the wrong direction. Well, it's not the only one, but it's one of the few. But this is where it comes into paltering. It is the case that if you make allowance statistically for the fact that these kids have got a big cerebellum, relatively speaking, this bit of the cerebellum called the vermis is small in relation to their very big cerebellum. So, they are paltering. They're basically saying these kids have disproportionately small vermal volume, but they're missing the point that, actually it's in relation to this very big cerebellum and it's completely inconsistent with all the rest of the literature. So people do this, but they don't, I think, do it intentionally, knowingly. In this case, this was really quite interesting because this was a paper using a mouse model of the autism. Yeah, I mean, there's all these autistic mice models with strange knockouts and so on. And the thing about that literature is that they all refer to this same small set of studies that were quite old. And you get the feeling they never go back and look at what's happened more recently in humans. And so I think this is why this sort of thing happens. But you get this sort of bias in the literature creeping in where people, I think, really just don't look widely enough. And so you see this in literature reviews, if you do look carefully, that there's massive misrepresentation, there's omission of things that don't fit, and there's paltering. And I think, though, that people don't regard this as terribly serious. When I gave this longer version of this talk, I had managed to get some sort of polling going on the phone where you get the audience to respond. I was going to try it here, but I decided I'd have a sort of nervous breakdown if I tried it. So, but basically, people do not regard this as nearly as bad as fraud, for example. And I think that there's good reasons for that. It's very hard to know if people are unintentionally leaving stuff out or deliberately leaving stuff out. And then typically, if you're writing a review for a paper in production, you have very limited space. You do not really want to spend a lot of time talking about all the stuff that doesn't agree. So you focus on the stuff that does. Everybody does this. And it's impossible to read everything. I mean, there's a huge literature in many of these areas. So you have to somehow select. And people think, well, there's no obvious victims. And they tend to think it's not really that much of a problem. I mean, it's not going to make a big difference, is it? Or is it? Let's have a look what happens. How big is the effect if you have biased reporting? Now, get your brains working before the coffee break. The Bayesians will love this. Everybody else will hate it. Suppose you've got a series of experiments testing effectiveness of a treatment. And we're going to say, if there's a significant difference, I'm afraid, yes, we are using statistical significance here, but you say it's a Y study, and if there's a non-significant difference, it's an N study, because you're just looking for a difference between a treatment group and a control group. And we're going to say that alpha is 0.05. So we know that if you get a positive result when there's really no effect, that's in 5% of occasions, and the probability of getting a null result when there's really no effect, 0.95 there. And then if it really is effective, we're going to say power is 0.8. So this is a highly powered study relative to the rest of the literature. And you can, again, you work out the percentage of likelihood of seeing a yes or a no when there really is an effect. And then from that, you can actually, provided you decide what your prior is, and we're going to say the 50-50 chance of working, you should be able to work out what's the relative likelihood of this treatment working or really having a genuine effect if you see a series of results like this, where you've got a positive, a negative, two negatives, and a positive. So what do you guys think? You don't have to tell me. I won't embarrass people, just in case you would feel embarrassed. But do you think this means that the treatment's very likely to be ineffective? It might be effective, but you need more experiments, or it's very likely to be effective. Have a think, form your impressions. Ta-da. So this is actually the log odds of a true effect shown in this graph, and it's actually suggesting that the treatment is very likely to be effective in the sense that the odds ratio, or the log odds of 3, indicating 20 times more likely to be effective than ineffective. So that looks like good evidence, and you may start to think, yeah, okay, we've had a few null findings, but this looks like it's a treatment that we should persist with. If we do the same exercise now with this sequence, what we've got here is a longer sequence of studies with more null results in there. And again, think this through. Would you think this is very likely to be ineffective or effective, but we're not quite sure? Or effective, we're fairly confident. Have a think. My guess is that I won't guess. I'll tell you afterwards what other people have said. But what you see here, of course, is it's now tracking in the opposite direction, down to leading to the conclusion, if you're going to say, by the time you get to a log odds of -3, you're thinking it's pretty likely to be a dud. It's just coming into that region. It gets even worse, because if you're p-hacking while you're doing all of this, this means that your effective alpha is not 0.05. Suppose you're looking at lots of outcomes and just picking the significant ones, like we showed you in the forking paths. Maybe your alpha is actually 0.2. And then if you do that, you get even lower, even more evidence against the real effect. So what we've actually shifted from is a situation where by omitting certain sorts of trials, because I haven't made that point clear, but the whole idea is that this second sequence is the same as the first sequence, but with null trials, some additional null trials that were suppressed, that were not looked at. So the idea is that if you actually omit half the trials that get null effect, you can completely shift. And it's the same really with citations. Now, this whole way of analysing things, I'm very grateful to Carl Bergstrom and his colleagues who published this beautiful paper in eLife, where they were talking about publication bias rather than citation bias. But I think it applies equally well to citation bias. And the idea is that what you do is, it's just as I've shown you here, you have a claim that may be true or false. You have evidence that supports it or fails to support it. And then you can either have a publication or not. And the point they make is you can get all these distortions and this sense that there's a very clear effect if you just fail to publish the null studies. So that's publication bias. But I would argue you can get a very similar effect if you just are failing to cite things, even if they get to the point of being published. If nobody then cites them, again, we build up this situation where we seem to think we've got an effect when we actually haven't. And this is really quite serious, the extent to which we use confirmation bias and other demands on our time and attention to only focus on things that fit our preconceived views. It's leading us, I think, I love this phrase, the canonization of false facts. I think our literatures are full of canonized false facts because we all do this all the time. And then we get what I call inheritance of bias. So we go from, we read a peer-reviewed paper, we tend to trust the citations. We can't read the entire literature on every paper we want to read. So we tend to trust them. But then we come to write up our own paper, we cite the same materials. If you're a good scientist and you do your best, you will actually read the papers you cite, but not everybody does. But if you do read them, you still may not be saved from this bias because you will inherit it. Again, you're not going to sort of read absolutely everything that's out there. You will rely on previous people citing the relevant literature. But if the relevant literature only cites studies that agree with a particular viewpoint, that viewpoint gets entrenched. And in the literature, as I found with these cerebellum mice studies, everybody, if you talk to people who work on the cerebellum in mice, they all think this is a very well-established fact. If you talk to people who work with kids, they don't think it is, but the literatures have diverged. And one literature keeps just citing the same old neuropathological studies that suggested that. So you don't know, unless you explicitly search, that there are other studies that give a different picture. So errors of omission and paltering in reviews can have very serious cumulative effects and a sort of false canonization of facts. And furthermore, this is not a victim-free crime. I mean, that's how people tend to think of it, that it doesn't matter if you do this because nobody gets hurt. But they do get hurt. If you're working in clinically relevant areas, you can get the general public will, in the long run, get hurt if ideas that are wrong about a disease or a condition or whatever are persisting through the literature because nobody really bothers to talk about the null results. Researchers, when we get these discussions in the psychology field about tone, coming back to Andrew Gelman's talk, and should you be rude to people, I have actually written a blog post about this because everybody was saying, oh, no, we can't tell other people that they're doing things wrong because they'll get upset. And the point is that I think we should tell them this because we've got a new generation coming through, and I keep meeting people who are early career researchers who have wasted 3 years trying to replicate something or build a new line of research on the literature, which they think is solid. It's not solid. And then, of course, the funders. The funders who, as far as I'm concerned, in the UK, our funders are pretty heroic because they're trying to grapple with this and make people be more reproducible in the research they do because they don't want to waste their money and they will have clout and they will cause these top-down effects. Now, can we solve this? Well, the clinical trials people have tried hard and they've said, well, let's try and overcome this bias. Let's do systematic reviews. Systematic review, you collect and summarize all the empirical evidence that fits pre-specified eligibility criteria. That's great. You pull it all together. But, as is well known, people say garbage in, garbage out. That's not the problem. The problem, again, is the stuff that you miss. Because if you do a systematic review, you first of all, if you're, again, a good, rigorous person, you collect abstracts and titles of papers to find the relevant papers. They only tend to mention the positive results. Here's an illustrative example which came out about a couple of months ago. It's a really terrible PHAC study. It's about solvents in the workplace and risk of autism spectrum disorders. It's a classic, when you read it, classic p-hacking. Big epidemiological thing. They looked at solvents, they looked at pesticides, they looked at exposure to this, exposure to that. They found one significant association, and they did have the grace to explain that it didn't survive the Bonferroni correction, but they were the sort of people that thought the Bonferroni correction was a sort of invention by nasty statisticians to make life difficult. So they nevertheless published it. And in the abstract, though, they just mentioned the solvents, which was the one significant result. Suppose you were doing a systematic review on autism and pesticides. You wouldn't find this study unless you combed the literature beyond this title and abstract, which is not what people can normally do. So it gets lost. So it's negative evidence, and it gets lost. So even systematic reviews are not a full solution, although they're certainly better than just relying on your memory for what's out there. So, Academy of Medical Sciences meeting where we came to these conclusions that we had problems, we need to change incentives, we need better training in methods. I think what's missing here is how humans think and reason, which we need to really understand in order to overcome some of these problems. I think there are problems which I didn't go into in this talk about understanding of statistics, which can be ameliorated by training, but there's also factors that will have to do with why people don't think it's wrong to do many of the things that they're currently doing. It's a mixture of misunderstanding, but also a tendency to think that it doesn't really make that much difference, and that omitting something isn't a very serious problem. I hope I have persuaded you that it is actually a very serious problem that we're going to need to grapple with if we're going to actually get our science to stop canonizing false facts and focusing on the much rarer true facts. Thank you.
Thank you so much for your moving talk. It's over here. But, you know, I kind of tend to think in analogies. And would it be fair to say that, say, we take a group of people, explain them the rules of American football, and let them play? And the main rule would be you need to get from here to there, get to the goal, whatever it takes. And then we start complaining, why are they hitting each other? Or why is all this mess? Why all these injuries? Why do they pay him $100,000 so one will knock out the other one without the referee saying anything, which is the fact of reality? Isn't it the situation? So scientists are set up in the environment where you get to get there, whatever the cost, or you're out.
Well, that's the cynical view of what science is about. Yeah, I get your point, okay, but that's the cynical view of what scientists are about. I would say most people who go into science because they actually want to do research and they want to find the truth. And it is unfortunate that our incentive systems have created a situation where, I mean, the other day I was not very kind about Darrell Bem, because Darrell Bem, famous psychologist, wrote this piece about how to succeed in psychology, which had exactly that type of attitude behind it. And in fact, there was a little blurb on the book, which was a book directed at early career researchers. And I think the phrase was, this book is going to teach you how to win the career game in psychology. You know, this is not what we're about. We're trying to find out what is the case. And it's about time that we just gave up on this idea that this is some sort of game that you play in order to succeed by getting lots of publications. It drives me insane.
But I mean, to correct the record, I agree that most scientists I know came to science to do exactly what you say. But this is my point. What if you take this type of people, you put them under conditions that are against their inner self.
I get your point.
Then you get, what we have, graduate students,
I think this is a potential explanation. Yeah. Anyway, next question.
Thanks for a really great, thought-provoking talk. Very engaging as well. I think this is a really deep problem you're getting at, and it ties in with the earlier talk about polarization as well, where there's different mechanisms that lead people to have access to different sources of information, and then it leads to stable differences in beliefs, and then people can't agree on something which we'd ideally like them to agree on. So I was more curious about your thoughts on solutions to this. So one of the thoughts you kind of shot down at the end, which was some source of systematic review. So I was thinking in medicine they have a Cochrane collaboration, which is supposed to be a independent group that does its best to do systematic reviews. And the nice thing about that, at least as far as I understand it, is that these people have less conflicts of interest, presumably, so they have less incentives to selectively gather information. So I was wondering whether you think something like that is feasible for the like social sciences, or if you've thought of other potential solutions to this type of
I wouldn't say don't ever try and do it. In fact, I would recommend that people do try and do a systematic review, because I think it still is probably likely to give you a more accurate idea of what's going on than just as I say, rely on people's memory or prior citations. And you can do it. You can do it in a way that should preclude conflict of interest, even if you do have some sort of interest in the field. There are guidelines that you can follow which require you to be, in fact, very upfront about what your criteria are for selecting papers and for judging papers and so on. The main problem is this business of the missing information that you may never be aware of. And I think the way that we might perhaps in future deal with that is more through, this is where data mining of text mining of papers could be more useful, because then you might pick up papers like this one that I'm mentioning, where you wouldn't know that they'd looked at pesticides from the abstract or the title, which is what people screen. But you could actually set up a text mining situation where you looked for mention of that anywhere in the paper. And if we had, with big data, ways of doing that much more rapidly, I think it could lead us to have more accurate representation of what is actually out there. I mean, we've still got publication bias that means that things don't actually even get into publication if they are null results. But nevertheless, that might be an improvement. And I think there are other ways that one can try and improve the reproducibility of science. I'm a great fan of registered reports and things like that. I mean, I think it isn't a one problem, one solution thing. But I was going to try and talk a bit about solutions, but I decided I wouldn't have time. But in this paper, I do talk a bit about solutions more generally.
Thanks very much.
Yes, it's more or less on the same sense. I mean, trying to tie up the two last lectures, I mean, the tragic thing about confirmation bias, it gets ever easier as science grows exponentially, right? We have like ever more data to cherry pick or cite from. And maybe tying up with like Kayleen's talk, if there's ever more data and less rigid data, you might be ever more mistrustful, and that messes up the whole thing. So, I mean, how do you counter that? Do you actually think systematic, like better methods of reviewing or detecting evidence might do, or do we actually need some kind of filter or progressive filters for rigor of evidence that can somehow stop the growth? Because like if there's more and more info, I mean, there's more noise, and it tends to get worse, right?
It does. And I mean, there is a very provocative view, which I've favoured for a very long time, but I think it would be impossible to implement, which is just to limit how much we can all publish. I mean, if we were all restricted to, say, 2 papers a year, the literature would improve markedly. So maybe the people here who are from Wellcome Trust, well, it's not their decision how many papers you publish, but I mean, they could, I suppose, impose it on the people they fund. That would be popular. Yeah.
So I think somewhat bootstrapping on that, I'm going to ask you to look at sort of the social commons and the culture of science. And in each of these examples where you have that distribution of results, there's actually a person or a lab and a voice behind each of those now ignored results, where the whole body of work has become entrained for the ones that people are following.
Yeah.
And in the social commons, there are a lot of studies around process and around communication process for being able to invite all the voices to the table. And I wonder why these other voices that have those less popular views that get left by the wayside, why we don't have a social commons or culture in science where we can hold a question with much more variance in what's on the table, so we can keep all of those present. And I think what I'd point to is a classic example of, I think it was the Challenger with the Thiacon O-ring, where there was one engineer who knew there was a problem with brittleness at the very low temperatures, and the writing over because of popular consensus finally had him be quiet. So this isn't quite that, but it's a version of that.
Yeah.
So I'm curious, and I guess my last question then, when you say, well, I'm going to be more forward about being abrupt and even, I forget how it was phrased before, but to be very direct with people, almost feels like it's going in the opposite direction to me, because it disinvites voices.
I think the voices that are, I see what you're saying, and I do. I mean, I have been talking to people here at this meeting about the conflict, the huge conflict I have. I was at another conference 2 weeks ago where I was going around a poster session as a senior person in a field where they haven't yet really quite woken up to the reproducibility crisis, and I felt that most of the posters were p-hack. So what do? Do I say nothing and be encouraging, or do I go around like the bully, sort of saying, well, you know, this is bad, or I can try and do it nicely and say, well, by the way, I do think this. But, you know, the fact of the matter is, I think we cannot be complicit. I mean, once you see that there are methods being adopted that are, I think science has to trump everything. It has to trump the social side of things and everything, getting at the truth. So I don't think we can say, you know, I shouldn't ever tell a graduate student that I think that they've got a problem methodologically with their poster or their paper because it will hurt their feelings or make them feel excluded. I think I should try and do it kindly and nicely and not in a way that will make them want to leave the field forever. But that's if we find something wrong. But you're talking more, or in addition, about the situation of the people who find the results that nobody's terribly interested in and things like that. And there, I think, we just, one of the reasons I wanted to talk today was to say we do have to, I think, recognise that this is not something we can fix by just sort of saying we have to have some policy to make this more, because it's a very, very deep psychological tendency in people that protects us from information overload if we just focus on the things that look really important and exciting and fit our current views. So we have to somehow, to be a good scientist, as people like Richard Feynman's famous for saying, you have to like bend over backwards to try and find the evidence that doesn't fit your view. And it's not a natural way of thinking. And we do have to start training people to do that and to listen to those voices of people who are saying things that they might not want to hear. But I think if we don't accept the fact that that needs you to work against your natural tendencies, we're never going to get there.
Hello. Hello. Daniel Acuna from Syracuse University. I work on trying to automatically detect falsification in scientific images with machine learning.
Oh, right.
So for a long time, I thought that people were just making mistakes and people were being pressured. But the more I saw the cases that I was discovering, I realized that a lot of these people were borderline mentally ill, just fabricating data, finding ways of making more sophisticated ways of essentially creating false science. So I'm wondering, so I was at a meeting on research integrity a couple of months ago, and I saw a big shift in how institutions were reacting to cases of falsifications and false science and misleading science after a big case of a Duke that essentially, basically because of fraud, a big fraud case, Duke got fined by NIH for $100 million. And I saw that all the institutions were essentially implementing a lot of measures, training, education on these issues. So what role do you think punishment has in trying to improve the practice of science?
I think that punishment will be probably one of the few things that will get the institutions to take action. And it's the institutions that do need to take action. So I think it's, if you worked in an institution where you were rewarded not for publishing lots of papers and bringing in lots of grant income, but you were rewarded for the extent to which your research met criteria for being reproducible and robust and so on, this would completely change everything. And that is a direction we are beginning to move into. I'm quite an optimistic person. Certainly in the UK, we have had some concerns at government level about research integrity, which has led all the universities in the UK to be sort of circulated with a requirement that they have to have a plan of how they're going to tackle exactly these sorts of issues. And they're all running around like headless chickens because it's so much at odds with all the other pressures on them to produce lots and lots of wonderful research that's very sort of high impactful and all the rest of it. But they are having to take it seriously because their funding will depend on it. So I think ultimately this is the sort of thing that can potentially be handled to some extent by top-down influences, and of course by training the bottom up in ethics and integrity. I think there's just, we've got a very corrupt scientific system in some places, because what happens is you get these, I've seen it happening in people I know, that you get idealistic people coming in wanting to, cure Parkinson's or something, and then they go and work in a lab where people say, no, we don't really do it. People think it should be like that, but that's not how it really is done. As if you're sort of, to be in this club and to be part of science, you have to sort of bend the rules and everything. And you're regarded as naive and crazily idealistic if you do anything else. I think that has to stop. And that you can, to some extent, if you start training people in ethics and warn them that if people start saying things like that to them, that the institution will be on their side and will not kick them out. But we're at a point of change, I think. So I'm optimistic that this will improve. But there's a whole generation of people who've been doing all these dodgy things for years who are very unhappy and are going to sort of be kicking and screaming.
Thank you for the wonderful talk. Zoltán Kekecs, psychology. I have been always advocating for the use of correction to correct for the inflated alpha error probability. But there is an argument against this for which I don't have a good answer. So when I have an argument with a department member about this, I always lose the argument because of one counter-argument, which is, okay, so I have, let's say I'm interested in the gene, candidate gene for a specific disease, and I have 10 candidate genes. I would test these in a single study, then I would have to use Bonferroni's correction.
Yeah.
But what if I decide to do 10 separate studies? So I could use the 0.05 p value or a p threshold at each of these studies separately. But if I take the 10 studies together, I still have an inflated chance of finding one of them falsely to be the responsible gene for a disease. So they would ask me, so should I apply a Bonferroni correction for my whole career? So all of the papers that I did so far, so with each paper that I do, should I decrease the p threshold? So what's the correct argument for that?
The correct argument is that you start with a hypothesis as to what you're predicting. And there may be, I think the whole career thing is a nonsense, but if what you're predicting is there may be a candidate, there may be one sort of SNP out there that is correlated with this phenotype. And if your hypothesis is you're going to look for a potential SNP in any one of 10 genes, then you Bonferroni correct. If your hypothesis is, I have a very strong prediction that COMT or something is the gene for this condition, you can set that up as an a priori hypothesis, and you can then not bother with the Bonferroni correction. But you've then made a very specific prediction about that specific gene. So it ties in with really just the fact that you have to a priori make a distinction between, are you testing for one particular thing, or are you looking for something is going on in this whole set of things, which requires a very different sort of approach to probability, I think. I mean, I'm not a statistician, and the statisticians here may disagree with that, but I think pre-registering is a big help, because if you do it honestly, because then you can be clear what your prediction was. And then I know people have said you don't always need replication, but in genetics, the general thing as well is that if you find something, if you are going to do it in this more exploratory fashion, then it's normative now in genetics that you would have to replicate. In fact, they've more or less given up on candidate genes because they find they don't replicate. But so they got wise to this problem precisely because there were so many comparisons you could do. But I don't think this is about, obviously you're not going to sort of Bonferroni correct for your whole career, but you are doing a study in a finite, defined way, and you should have a hypothesis that you're testing, and it should be clear whether that hypothesis is this specific thing is going on or something is going on, in which case you're doing something more exploratory and you need a bigger correction, and you should then replicate. That's how I would respond to that.
Okay, I think they would say, okay, but what if I come up with the next hypothesis after? Yeah, thank you for your answer. But I think that's I will still talk to some statisticians. Maybe
we should talk to Andrew or something. I mean, I think it's a slippery. Probabilities are very slippery things, but ultimately, people do these studies where they do a whole genome thing, and then they do just a big plot of the obtained probability against the expected probability for the whole distribution, so you can see if things are sort of falling off that GG plot, that QQ plot that you have there.
Leif Nelson, I'm a psychologist. So very compelling narrative about the role of confirmation bias, and not surprisingly, I totally agree with all of the observations you've made. Something that's interesting, and as I'd say, is the difference between p-hacking and, for simplicity, citation hacking, as you describe it, and that when a researcher p-hacks their finding, they have all of the confirmation bias working in one direction, but also there's no correction mechanism possible. That is, I'm the only person that knows about the analyses that I'm not reporting. But I would have said with citation hacking, there's a whole field with deep incentives to prove me wrong. If I say, oh, the literature all agrees on X, here's 4 citations, there's an entire Twitter universe of people that want to say, ooh, Nelson didn't mention these other ones over here.
Yep.
And so I'm curious, given those pressures, why is it the case that we don't see instantaneous, substantial, and I would say even totally effective countermeasures in that world?
Well, there's a good point about social media, because social media, of course, has only been around doing this sort of thing in the past. I think academic Twitter, for example, hasn't really come into its own except in maybe the past 5 or 6 years. So I think this may start to be on the increase. But and you're right. I mean, ultimately, this is detectable in a way that p-hacking is not. I hadn't thought of calling it citation hacking, but I guess that you could call it that. I mean, it is just sort of omitting the things you don't like. So it is ultimately detectable. It's just that I think for a lot of these things, you get these vogue areas which get up ahead of steam. I mean, we heard about, the oxytocin story the other day. So there's always a few people, typically like me or Andrew or somebody, who sort of are sitting there going, yeah, I don't believe that. But most, you get these ideas where everybody's rushing onto the bandwagon and everybody only wants to see the positive stuff. And then you get the hype cycle. It goes, and then it comes crashing down when other people eventually can't replicate it. But in the past, this whole hype cycle would take about 25 years. And it would waste a lot of people's careers while they tried to chase the bandwagon. So I think it's possible that, it may, I hope we can find a way to speed it up, but perhaps by being more rigorous in how we review literature reviews, and more rigorous in just sort of, getting onto Twitter and perhaps being bold enough to say, hey, you didn't cite my work, or this study. So I think things will change. But I think the real problem, there was a beautiful blog post by Slate Star Codex. This guy got a blog about how long it had taken before they realised that one of these genetic associations with depression doesn't stand up in a huge meta-analysis. And he wrote very eloquently and quite funnily about how long it had taken and how many studies it had taken before we came to the conclusion that the association was not there, because there was study after study after study doing this. And it was about 25 years' worth of studies, huge amounts of research dollars, huge numbers of people's careers, I think, sort of down the toilet, probably lots of people with depression getting given peculiar drugs. So, the consequences were fairly major. And it should be that these things may happen, but the correction cycle should be much, much quicker than it currently is. And I think maybe social media may turn out to be a mechanism for that. But at the moment, if you do go onto social media and start, chayacking at what people are doing, you will get the kickback that you're a very nasty, bullying person. I get less because I'm a woman, but there's a lot of people that will say, oh, it's these nasty men getting at us because, they're bullying men. And I think we just have to say, well, science is about getting it right. And, it's about being not attacking the individual, but certainly looking very rigorously and critically at the idea and not trying to continually prove your idea, but rather you yourself should be trying to disprove your idea. We seem to have got away from that as a way of thinking about how to do science.
Thanks.
David Allison, Indiana University. I wanted to make a point and ask your input on something that I think has been glossed over several times in this talk and yesterday a little bit in some of the Q&A after Andrew Gelman's talk. And I think what's being conflated sometimes is silence with politeness. And I'm hearing people say we shouldn't be polite, we should offer criticisms when criticisms are due. And I don't think those are two different things. I think we can and should be highly critical of methodologic errors and issues, just as you have said a few moments ago. I think we should assertively, crisply, and publicly point out when something has not been done in the literature correctly. None of that has anything to do with being impolite. I think there are moral as well as practical reasons why when we offer our criticisms, we should do so professionally, civilly, and politely so that we are not seen as bullies, so that we don't unduly penalize someone who made an honest mistake, and so that we are not offering people who make mistakes the opportunity to just put us off by saying, oh, you're just a bully with a personal vendetta. So I really think it's important that we not conflate politeness with silence, which has been done in this session.
Thank you. I completely agree. I've actually written a paper on fallibility in science and what you do when you find errors in your own work and in the work of others, where I try to make a very similar point. I think the difficulty, though, is that there have been some voices in psychology, certainly, who have basically implied that if you say anything negative about somebody's work, you may be damaging their career. and that therefore you shouldn't say anything negative about somebody's work, which is making that person's career, I mean, it shouldn't be damaging their career. My whole point would be that errors are normative. And the other thing that senior people like me can do, and I have done on occasion, Russ Poldrack, who's here at Stanford, has been very good at saying this, is that we all make mistakes. We all are fallible. And that it is very important that when you make a mistake and you don't sort of bury it, but you should actually be as public as possible with it, so that we get an attitude going around that this is a normal part of science and it's a normal part of humans to just screw up or to not understand something, and that it shouldn't be a source of shame. But we're not at a vicarage tea party. We're trying to do science. And
Thank you. If I ever get to a
Thank
vicarage
you
tea
for your
party and
question.
learn what it is, sounds great.